Welcome to Biotanemy.
Hello guys, this video is a continuation from two previous videos in our channel, Gel Electrophoresis and PCR. In the previous videos we discussed about the basics of Gel Electrophoresis and PCR. In this video we'll see Pulsed-Field Gel Electrophoresis or PFGE and different types of PCR. First, let's see what is PFGE. PFGE is an electrophoretic technique that is used to separate DNA molecules larger than 50 kb to more than 10 megabits. It is commonly employed in microbial genotyping and other large-scale genomic studies. The procedure for PFGE is 1. The microbial cells are embedded in 1% to 2% agarose plugs to protect the DNA from shearing during handling. 2. Lyse the cells within the plugs to release intact chromosome DNA, using Lysis buffer. 3. The DNA is digested with restriction enzymes to produce large fragments for analysis. Next step is the electrophoresis or PFGE and visualization. Types of PFGE are 1. Standard PFGE. Here the electric field will alter the direction at fixed angles of 120 degrees. Is used to separate large DNA fragment. 2. Rotating gel electrophoresis. The gel itself is rotated during electrophoresis to allow uniform reorientation of DNA. It will provide better resolution for very large DNA fragments of more than 2 megabits. 3. Field-Inversion Gel Electrophoresis. Here the polarity of the electric field is varied forward and backward direction. This method is used to separate 10 to 500 kb DNA fragments. 4. Transverse alternating field electrophoresis. The electric field will varies in 90 degrees angle. This will separate the DNA faster with high resolution. 5. Contour-clamped homogeneous electric field. This method uses multiple electrodes arranged in a hexagonal pattern to generate a uniform electric field. 6. Orthogonal-Field Alteration Gel Electrophoresis. The electric field alternates orthogonally at 90 degrees angle. It is used to separate large DNA fragment. PFGE is used for microbial typing, genomics, and food safety. The limitation of PFGE are: they are time-consuming and labor-intensive. While normal electrophoresis takes few hours to complete, the PFGE will take few days, and it requires specialized equipment and expertise. Now, let's move on to different types of PCR. Different types of PCR is done in order to overcome few problems encountered during the process. Some of them are: 1. Nested PCR. Nested PCR is a highly specific and sensitive modification of conventional PCR that involves 2 rounds of amplification using 2 sets of primers. The first round of PCR done normally with 2 sets of primer. The next round will incorporate a pair of different primer, which binds with the sequences inside the product of the first PCR round. It will improve specificity, sensitivity. Also reduce the background noise or unwanted sequences. 2. Hot start PCR. Hot start PCR is a modification of conventional PCR that prevents nonspecific amplification and primer-dimer formation by keeping the DNA polymerase inactive at low temperatures. In regular PCR, primers can bind to non-target regions at room temperature, leading to unwanted amplification. Hot start PCR avoids this by keeping the polymerase inactive until the reaction reaches the denaturation step. It can be done by using antibody to block the polymerase, which will be removed by high temperature during denaturation. Or polymerase can be covalently blocked using heat-label blocking group, like reversible thiol group. Some hot start PCR use chelating agents like EDTA to temporarily chelate magnesium ion, which is essential for polymerase activity. Some physical barrier like wax is also used to separate reagents at room temperature, which will melt away at denaturation temperature to start the reaction. 3. Error-prone PCR. Error-prone PCR is a modified PCR technique designed to introduce random mutations into a DNA sequence by reducing the fidelity of DNA polymerase during amplification. It is commonly used in directed evolution, protein engineering, and mutagenesis studies. It can be done by using unbalanced dNTP concentration, addition of manganese, or use of higher amount of magnesium. 4. Real-time PCR. Real-time PCR, also called quantitative PCR, is a technique used to amplify and simultaneously quantify DNA during the PCR process. Used in gene expression studies. Unlike conventional PCR, which detects DNA at the end of the reaction, qPCR monitors DNA amplification in real time. The amplification of DNA is analyzed using a fluorescent dye or probe by measuring DNA quantity after each cycle. The fluorescence intensity increases proportionally to the amount of amplified DNA. SYBR green dye is used in fluorescence-based detection. SYBR dye will intercalates the double-stranded DNA to produce fluorescence. One limitation of this procedure is it will non-specifically binds with all dsDNA, including primer dimers. In probe-based PCR, it used sequence-specific fluorescent probe. For example, TACMAN probes, molecular beacons, Scorpion probes, they have a fluorophore and a quencher. The fluorescence produced by fluorophore is quenched by the quencher. When the DNA polymerase extends the strand, the quencher is cleaved, separating it from the fluorophore, which will allow the fluorescence to emit. Another technique we can use is the FRET or fluorescence resonance energy transfer. FRET use two probes, donor probe and acceptor probe. Donor probe are designed to bind near the 5' end of the newly formed target strand, and the acceptor probe to bind near 3' end. When they adhere together, the donor fluorophore absorbs light, and the energy is transferred to the acceptor fluorophore via FRET if they are in close proximity. The acceptor fluorophore then emits fluorescence at a longer wavelength, which is measured in real time during the PCR amplification. So, that's all are the different PCR techniques used in molecular biology. If you like the video, please click on like. Thank you.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.